It's my great pleasure to introduce Lucas. Lucas Majure is a botanist originally from Mississippi. He earned a Bachelor's of Science and a Master's of Science in Biology from Mississippi State University and a PhD in Botany at the University of Florida in 2012. He's a plant systemicist and evolutionary biologist and focuses on the understanding of the evolution and maintenance of plant biodiversity. He especially focuses on the Caribbean and Andean South America and arid and montane ecosystems throughout the Americas. Major groups of plants that he studies are the cacti, particularly opuntiads. First time I met Lucas many years ago was because of a joint fascination by prickly pears. And the plant families Melostomataceae, princess flowers, Poaceae grasses, and Cimarubaceae, the tree of heaven family. So he's going to be talking about Melocacti of the Caribbean, the how, the when, and the where of their arrival in the islands.
And with that, I'm going to mute myself, turn off my video, and hand it over to Lucas. You should be able to share your screen.
All right, I'm gonna share right now and make sure everyone can see it. Let's see, can everyone see my screen? And can everyone hear me all right?
Yep.
Yes.
Excellent. Well, I want to start off by saying it's a really great pleasure for me to be here. So I want to thank you so very much for inviting me to talk with you today. Especially talk with you about a part of the world that I really love, a part of the world that's just absolutely loaded with biodiversity. And I think this group, Melocactus in the Caribbean, is a really good example of that type of biodiversity that you can find in a place like the Caribbean. And so, so with that, I guess I'll start off with this first slide that I wanted to show you. This was back in 2018 when we were doing work across the island of Cuba. And this is a couple of colleagues of mine here that are in the front part of the photo, Duniel Barrios and Edgardo Diaz. And this was one of the most amazing scenes I had ever encountered in terms of numbers of individuals of Melocactus. This is the species Melocactus harlowi from the Guantanamo area of Cuba. And an absolutely phenomenal place to see species of Melocacti. And, Cuba is one of those places that has, as I'll show you a little bit later, one of the highest numbers of species specifically within the Caribbean. So let's go ahead and start off just by orienting ourselves a little bit. I mean, I'm sure that everyone is familiar with the Caribbean region, but I always like to just throw out a few things about it. So one thing that's really curious and really fascinating about the Caribbean is it's one of the top 5 biodiversity hotspots on the planet. And what that means is that, there's a relatively small area, but for the area that makes up the Caribbean region, there's a very high number of species that occur there, and there's a very high number of species that occur there and nowhere else across the planet. So the level of endemicity is incredibly high and upwards of 50% in some cases. And cacti on some of the islands, as I'll show you in a little bit, that level of endemicity might be 70, 80%. And so it's an incredibly interesting area from the standpoint of plant evolution and trying to understand how these areas have been occupied by plants, how those plant communities have developed over hundreds of thousands to millions of years. And two of those that are really important places for me especially are the islands of Cuba and Hispaniola. So the islands of Cuba and Hispaniola are the largest islands of what we call the Greater Antilles. So the Greater Antilles being composed of Cuba, Hispaniola, Puerto Rico, Jamaica, Cayman Islands, and the U.S. Virgin Islands. Those are all on the same tectonic plate. And these two islands are very special. They have a really amazing geologic history. They're the largest within the Greater Antilles, but they also have the highest diversity of any of the other islands throughout the Caribbean and specifically within the Greater Antilles. And it's a really special place for us, especially for what we're going to talk about today, because it's both of those islands have lots of seasonally dry tropical forests. So these are, relatively arid forest, specifically within certain times of the year. Most of these are on either serpentine soils or limestone soils, sometimes silty soils. And it's an absolute perfect place for groups like cacti to exist. And within the Caribbean region as a whole, there are almost 100 species of cacti that are known throughout the Caribbean. Many of these, of course, of which are endemics or only known from the Caribbean region itself. But a lot of those numbers are actually the result of all of the diversification, especially in situ diversification that has happened on the islands of Cuba and Hispaniola. And so just to sort of give you a really brief overview of what you might expect to find on these islands, and I'm sure a lot of people listening today are already super familiar with this, but over the past 10 or so years, we've been doing a lot of field work. We've been really trying to understand exactly what species occur on which islands, which species, how do you distinguish the species one from the other. So we've been really working on the taxonomy of these groups. We've recently published this annotated checklist of the cacti of Cuba. So we found there are roughly 49 species of cacti that occur on Cuba. Those number is actually higher if you include the non-native species, but you'll see some common patterns here. I'm going to compare this with Hispaniola in just a second, but we see the genera Melocacti, Cylindropuntia, Hericea, Leptocereus, Opuntia, Consolea. If we switch to the island of Hispaniola, which we've also published a checklist for relatively recently, again, we see Opuntia, we see Melocactus, Mammillaria, which is also in Cuba, we see Cylindropuntia again, Consolea, Leptocereus. So the really interesting aspect of this is that, if you're thinking about cacti and we know that they originated on the American continent, most likely in Southern South America, and from then they've dispersed sort of all over the Americas. And so when you see these really disparate groups, Opuntia, Mammillaria, Melocactus, all of these things that are not related to one another, all in one place, it brings about the question, okay, how did they get there? When did they get there? So what were the means by which they got there? How did they do it? And when did they do it, right? And those are some of the questions that I'm going to be posing today with regards to Melocactus. I did want to put a shout out there to the importance of field work that we do. So these are photos of myself and collaborators on both Cuba and Hispaniola, and we do a lot of field work, and we generate a lot of herbarium specimens. And these herbarium specimens that we generate are essentially the backbone of all the rest of the work that we do. So all of the, I'll be showing you some phylogenetic trees in a little bit, showing you the evolutionary history of the plants that we study. But the backbone of all of that data that we generated, be it pollination data, data on morphology, data on genetics, anything like that, they're all backed up by these herbarium specimens. And the importance to that is just making sure that we make our science repeatable, right? So if Mitch says, I don't know if you actually sampled, Melocactus ebi, I can tell him, well, go look at the specimen and see for yourself, right? And so it's a really good way of making our science repeatable. And so these are some of the examples of the herbarium specimens that we make. I'm the curator of the University of Florida Herbarium, and so these are the specimens that are really, this is a very small example, but the specimens that really form this backbone of any of our research programs, right? One other thing I do want to mention about this is, because a lot of us grow these plants and, you see something dried and cut up and you think, oh, it kind of just breaks your heart. Well, when we're out there working in populations of these plants, we make sure that we're very careful not to endanger the populations. Oftentimes for things like Melocacti, instead of slicing open and drying a really beautiful, healthy plant, we'll take something that's been, it's fallen off the cliff, or something that's been knocked over by road work, or maybe one that's been damaged by a hurricane or something like that. And so we do our very best to make sure that we're not endangering any of these populations of the cacti we study in any way. So this kind of gives you an overview of what you would expect in the Greater and Lesser Antilles, in the Caribbean region as a whole, when you're thinking about diversity of cacti and the numbers of species. And this is not everything. This is just sort of a cross section that I wanted to show you quickly. So we have the genus Consolea of about 9 species, and those are Greater Antilles, Lesser Antilles. Genus Leuenbergeria, which is essentially restricted to Hispaniola and Cuba. Melocactus, 7 to 18 species, right? And that's something I'm going to dig into in a little bit later. So there's a lot of taxonomic, or was a lot of taxonomic uncertainty around the species of Melocacti in the Caribbean. We have Leptocereus of about 20 species, Pilosocereus 9 to 12, Opuntia with 12 or 14 species, Horacia with about 9 species. So these are some of the principal groups that I wanted to show you, because they're groups also that have diversified greatly within the Greater Antilles as compared to some of the other genera that don't really have a lot of high diversity within the islands. So, so now let's think a little bit about Caribbean biogeography. This is something that people have been wondering about for, literally hundreds of years. So if you're out in the middle of the ocean, how do these plant communities originate? Where do they originate from? There's been a lot of speculation. There's been a lot of analyses based on plants, based on animals, and other organismal groups to try to get at this answer, right? We know that the Greater Antilles really started out in what is now the Pacific Ocean. It kind of went under the continent before the Isthmus of Panama was totally sealed, and it ended up in the position it's in at the moment, roughly 60 million years ago, something like that. And then the islands that make up the Caribbean region were several different sets of different islands, especially Cuba and Hispaniola, and then those accreted together during the Miocene about 15 million years ago. And so since that time, we've had roughly the confirmation of what we think are the modern day, Greater Antillean and Lesser Antillean islands. And the Lesser Antilles was essentially this volcanic arc, which popped up. But one of these scenarios that has been posited for how things got into the Caribbean, say from continental, South America, is this Garlandia hypothesis, which essentially was a land bridge that formed from northern South America all the way up into the Greater Antilles. And this is supposed to have happened around 32, 33, 34 million years ago, something like that. And that's going to be really important when we're thinking about the family Cactaceae, because the family Cactaceae, as far as, at least based on our estimates, is probably around 35 million years ago. It's a really young family compared to most of the other flowering plant families. And so this Garlandia hypothesis does not really work for us when we're trying to make decisions or provide hypotheses about how this group ended up in the islands. And so what we've been doing is developing these data sets to try to determine, okay, what are the ages of those groups that occur in the Greater Antilles? So that gives us an idea of when they started to diversify, maybe when they could have been dispersed long distance from a South American ancestor, a North American ancestor, a Central American ancestor, et cetera. And one of those that we've worked on in detail is the genus Consolea. Essentially, we found, I'll just make it short and sweet, but what we found with this group is it has, close relatives in South America. It's a group that probably the ancestor of this group originated either on Cuba or Hispaniola before expanding across onto other islands. And it's super, super young, about 500,000 years old, maybe up to 1 million years old. And so in, geological terms and plant evolutionary times, that's really, really young. And it seems that this group has speciated because of the changing expansion and contraction of tropical dry forests during the Pleistocene, which was a major series of events that happened all across the islands as well as across the world. What about Leptocereus? We have this in situ radiation. So this is a group of about 20 species. Most are on Cuba. There are quite a few on Hispaniola. There's some on Puerto Rico as well. Group of about 20 species. What can we learn by understanding the evolutionary relationships of this group? The interesting thing is this is a group that you can tie directly back to the Andes because it's very closely related to the genus Armatocereus, which you find in Peru, Bolivia, and so it's one of these groups that's pretty easy to tie back to the Andes. So Daniel Barrios, colleague of mine, put together this phylogeny and showed that there were several main groups. There's essentially a group that's specific to Cuba, a group that's more or less specific to Hispaniola, Puerto Rico, and then this group that kind of spans both of those, which we used to call the genus Dendrocereus, that's totally nested within Leptocereus, and so we combine those into Leptocereus. But this is a group that's about 5 million years old, so a little bit older than Consolea, and so it's had ample time to speciate and to really develop into a large number of species across the islands. Alan Frank published a nice paper in 2013 where he looked at Hericea, since that's another one of these large genera across the Caribbean. Also has South American ancestry. It's very clear. And then here we have, probably a Pleistocene origin. So it's also a relatively young group. So we'll start to see this pattern over and over again, that most of the cacti that we see in the islands are super young, right? Even those in situ radiations that have a high number of species. The genus Apuntia, one of my favorites. This is one that, there are roughly, let's say, 12 to 14 species that occur within the Caribbean. Those species that are endemic to the Caribbean are part of a group that are also super young, probably Pleistocene in age. So we're talking a million or 2 years at the absolute most in terms of the age of those species. Pilosocereus, just very, very quickly. Pilosocereus, incredible distribution, right? With a lot of diversity in eastern Brazil. A lot of diversity within the Antilles as well. I wanted to show you Pilosocereus because it does mirror somewhat what we see in Melocacti, which is really interesting. But if we look at the phylogeny that was produced by Laborde et al. In 2018, you can see that, hopefully you can see that on the right-hand side of my screen with this phylogeny here. The Caribbean species of Pilosocereus fall into a couple of different places within this evolutionary tree. And what that tells us is that this group of plants has made it into the Caribbean more than once. In this phylogeny, you could say it's made it twice, right? But if you were to include all the Pilosocereus, who knows? Maybe they've made it there 4 or 5 or 6 times. Hopefully we'll figure that out at some point. Okay, so now let's look at the distribution of Melocacti. And this is a map that I put together a couple of years ago. We have more points for these now. But you have, and I don't have a point in Mexico, which I should, because Melocactus curvispinus occurs in Mexico, and Melocactus caroli linnae occurs in Jamaica. But what you can essentially see here is this, broad distribution of points in eastern Brazil, because that's one of the centers of diversity of this genus. And then you see, points scattered throughout the Caribbean, and quite a number of points on the island of Cuba, because that's another center of diversity. And so in Cuba, this number gets thrown around. And actually, I have 7 to 12 species, but the number that has historically been thrown around is about 3 to 13, something like that, in Cuba. And so this is, we were really interested in trying to tease apart, first of all, what are the species there? And then how many times did this group Melocactus make it to the Caribbean in general? And then when did that happen, right? So just to throw in a couple of other studies that have been done in the genus Melocactus, because there are some interesting things that happen in Melocactus that also happen in groups like prickly pears, the genus Apuntia. And one of those is polyploidy, which is also known as whole genome duplication. So this is a situation where instead of having the base number of chromosomes as a typical diploid would, say 22, and that's what it would be for cacti, they might have 44 or 66 or 88. So they have these essentially duplicated numbers of chromosomes. And why that's important is because oftentimes when you have duplicated chromosomes, it shows that there could have been hybridization in the history of that species, right? So if we look at some of these Brazilian species, Melocactus bahiensis, Conoideus, ernestii, they have that duplicated genome. And so probably what that means is those species could have been derived from hybridization between, one species and another species that hybridized naturally. They crossed to form that hybrid, and then the hybrid doubled its chromosomes, so it could actually, those chromosomes could pair and it could become fertile again, right? So this is something that happens really commonly in Melocactus. And so it could give us sort of a hint at how Melocactus has been diversifying throughout its evolutionary history. And then there have been some population genetic studies. So on the right-hand side of the screen, you'll see these figures that are sort of highly colored in yellow and green and purple. And what that essentially is just showing that there was hybridization between Melocactus glossescens, Melocactus consinus, and Paucispinus. So in populations where these things occur together, they're actually hybridizing with one another. So they're, maybe you have a hummingbird that goes to one flower of one species, then it goes to another flower of another species, and you get these crosses that happen. So this seems to be a pretty common thing that happens in Melocactus. Some other really cool things about Melocacti that make them interesting from a biological point of view, and one of those is many of the species are selfing, which means that they flower, and if that flower closes up even without pollination, the pollen is going to fertilize the flower and you're going to get fruit formation. Some of these Antillean Melocacti, I grow them in my greenhouse. They flower with no pollination whatsoever, and then they form fruit, and that's because they're selfing, right? So there are quite a few species that do that. They're typically hummingbird pollinated, but there are some other insects like honeybees and things like that that will go to the flowers. It's really curious that a high number of these species, so this has been recorded in Cuba as well. This one that I'm giving you an example of, La Cencianas, is from Brazil. And it's really curious that lizards are oftentimes the one dispersing these things. So what that also means is, probably you can get long distance dispersal because birds are picking up these really brightly colored fruit and moving them longer distances. But there's probably a lot of short distance dispersal that happens. And what that could mean from, say, a genetic point of view is you might see a lot of localized genetic structure. And that's something that might make a difference when we look at some of our data in just a moment. So what about conservation? There are a lot of species of Melocacti that are endangered. This is a pretty common sight that you see in certain parts of the Dominican Republic. So people there, and there could be some reality to this, but people think that Melocacti are really good for renal problems, so kidney problems, and they'll make teas out of those, drink the teas, and they use them for other medical reasons as well. But there's a lot of extraction of these for these types of purposes, and over collecting and things like that as well. And so habitat destruction is another one of those. So this is a group that's really, really important for us to learn more about so we can also understand how to conserve them, right? So there's some previous phylogenetic work in this group that gave us an idea of the most closest relatives to Melocactus, and one of those was Hernandez et al. In 2011, who included Arrojadoa and Discocactus, and showed that Discocactus was a likely sister group to Melocactus, which makes perfect sense. You have this really high diversity in eastern Brazil, and then that's part of the range of Discocactus as well. And morphologically, they look very similar. Tanya did find a recent divergent time during the Pleistocene for this group. So that kind of gives us a hint that this could be a really young group of plants. Taylor et al. In 2014 mentioned in one of his papers that there was no phylogenetic resolution among Brazilian species. And so it seemed like it was going to be really difficult to tease apart relationships using phylogenetic data. More recently, there have been some plastomes assembled, and what that means is these are the chloroplast genomes that have been put together in a number of different ways. And so there's been more and more data that people have been generating for this group of plants. And that's really, really good for us because the more data we have, the more we can try to tease apart relationships. And so that's what we set out to do with trying to understand the Melocacti of the Antilles. These are three collaborators on this project: Daniel Barrios from the University of Havana, Dr. Julay Encarnacion, who's a former grad student in my lab, and then Edgardo Diaz, who also helped us throughout Cuba working on these plants. So in this photographic plate that I have for you here, it kind of gives you an idea what a lot of these plants look like. Some seem to be sort of iterations on a common theme, and others are really, really different, right? I'm not going to go through each one of these because that might take a little too much time, but let's just pick out a couple of these. So look at A and B. We can see two taxa that look quite a lot alike. So A is Melocactus intortus, which is really widespread throughout the Lesser Antilles, makes it into Puerto Rico and the Bahamas. B is what we call Melocactus pedanalensis, which looks a lot like entortus, and some people have thought that it could be the same thing. And then let's look at G. If we compare G with the rest of the plants on this plate, it looks really, really different. You can't tell the size here, but that's a really, really tiny plant. This is Melocactus matanzanus, the famous Melocactus matanzanus in habitat in Cuba. And it really looks a lot like some of the Northern South American or Brazilian species that you see. And so, which is really curious. So from a morphological standpoint, I see something like that and I go, I think it's probably more closely related to something in South America. And that had been posited by Nigel Taylor previously, actually, because of its morphology. Most of the rest of the taxa you see here are relatively, sort of iterations on a common thing. We do have C and F here, which are Curvaspinus and Olguinensis. Actually, C was given another name, but anyway, I'll get to that in just a moment. So what about species limits in the Melocactus of the Caribbean? We have been working on some seed morphology to see what that might be able to tell us in terms of species limits within this group. And so that's something that Lemos Barrios published earlier on. And then we decided to tackle some of these other issues. So in Nigel Taylor's monograph of the genus Melocactus, he didn't really tackle the Caribbean that much. That was kind of, left off. He recognized 3 species from Cuba, a really broadly circumscribed Melocactus harlowi, Melocactus curvaspinus, and then Melocactus matanzanus, which is the one that I've got pictured right here. Melocactus guittarti and olguinensis were placed in the Melocactus curvaspinus because they have a morphology that's quite similar to curvaspinus, really big, giant red fruit too. Melocactus matanzanus, we had an issue there. There was a species described called Melocactus actinacanthus by Alberto Areces, and looked a lot like matanzanus but had fewer spines. And then we had another taxonomic issue, Melocactus pedanalensis, which was described by Mejia Garcia from southwestern Dominican Republic. And Areces thought that was more closely related to entortus, and so he put it within entortus. And then also Lemari and Prerepticula were 2 other names that we wanted to try to tease apart. What are those really species, or were they just morphological variants or whatever? And so here were our questions. We wanted to see if we could use whole chloroplast genomes to resolve the species relationships in Caribbean species of Melocacti. I was a little dubious about this at first based on some things that Taylor had published early on, saying that, resolution is really, really bad in this group of plants. Well, with this phylogeny, we wanted to determine were the Melocacti of the Caribbean derived from one radiation or multiple clades. So that means, for instance, Leptocerus, we know that was essentially one introduction into the Greater Antilles, and then they diversified after that introduction. And for the Melocacti, if you look at them morphologically, you can see there are iterations on a common theme for some of those, but then you have something like matanzanus, and then the curvaspinus group, you go, probably they made it to the Antilles multiple times, right? So that was our hypothesis. And we also wanted to know, how can this phylogenetic tree tell us or inform us about the overall ages of this group, and then the overall biogeographic history. We presume that this group would have a South American origin based on, the broad distribution and a lot of the diversity in Eastern Brazil, but these were all things that we wanted to try to figure out. And so we sampled 26 taxa. These are all from the Antilles. And then this was taxa that we sampled from fieldwork, herbarium specimens, and then some live specimens from DBG and actually Huntington Botanical Garden. We used a specific method for some of these older herbarium specimens where we had almost no DNA. We used this thing called rolling circle amplification, which essentially just makes many copies of whatever small amount of DNA you have in the DNA extraction. We had to do that for some of these which had very poor DNA or quantity of DNA for some of the older herbarium specimens. And then essentially we used this system where we generated millions and millions of short 150 base pair reads from different genomes from these species. And then I took those millions of reads and I assembled those to a chloroplast genome. So essentially what that does is pulls out all the chloroplast genome data, and then you generate a data set from that chloroplast genome data and then run analyses on it to understand the evolutionary history. And so this is what we get. And I know this is not something that's terribly easy to read. So I'm going to highlight a few things here for us. This is our phylogenetic tree that we derive from these data. So lo and behold, we have a whole group of species that are from Cuba. So we call this the Cuban clade. So this contains things like Melocactus evi, Melocactus acunai, Radoxi, Harlowi, and a couple of other taxa. And I'll dig into that a little more in just a moment. We had a South American group. So this is Melocactus ernesti, aureus, conoideus, bahiensis. Oh, and lo and behold, Actinocanthus and Matanzanus fall within this group, which morphologically makes a lot of sense. They look a lot like the South American taxa. And then we had this more broader Caribbean clade. We called it the Caribbean clade, although we do have two Peruvian species that fall within this clade, Beavistensis and Peruvianus. We also have the super broad, widespread Curvaspinus. And actually, one of the individuals of Curvaspinus that I used here was from Panama. The type of Curvaspinus is from southern Mexico. So this is something that's known from the continent. And then it's also known from the sort of northern South America and the southern Lesser Antilles. Taxa such as Caroli linai, Guittardi, Oleginensis that occur on Cuba are very, clearly related to this group morphologically, and they happen to fall within this clade as you would expect. And Tortoise also falls within this clade. And Tortoise, which is the largest species of Melocactus out there. So when we look at the overall biogeographic history and the divergence times, or that's to say the age of these groups, what do we see? So we see what appears to be a very clear South American origin of this group as a whole. But then we have multiple dispersals from South America to Cuba and or other parts of the Caribbean. We also recover a super young age for the genus Melocactus as a whole, about 1.1 million years. And again, there's a little bit of error that's associated with these estimates that we make, and so maybe it's 1.1 million years plus or minus 500,000 years, something like that. But that's still suggesting a very, very young age for this group, and that's Melocactus as a whole. If we dig into those groups that occur within the Antilles, they're even younger than that, right? So we're looking at ages that are actually kind of close to what we found with Consolea. And so here in this kind of this crazy, half circle tree that I have here, if you see anything in yellow, that's something that occurs on Cuba. And if you say anything in yellow on the backbone of the tree, that's an inference of an ancestor from Cuba, meaning that would have been a dispersal from South America into Cuba. So we have dispersals from South America into Cuba. We have Cuba to Hispaniola. And then we have these long distance dispersals from potentially from South America or northern South America, Lesser Antilles into Jamaica for Caroli linai. So what this is showing is that Melocactus has arrived in the Antilles multiple times, which is not a huge surprise given the type of fruit they are. They are bird dispersed to some degree as well. And so if you had long distance dispersal by birds, you would expect to see this type of pattern. And we do see this in other groups of Caribbean plants that have berry fruit. Oftentimes there have been multiple dispersals of those groups into the Antilles. So the South American and Caribbean clade here, just to sort of go into a little bit more detail, what we see with regards to the Cuban species is they fall out kind of all over the tree, right? So we've got Actinocanthus matanzanus, which now we actually think are the same species. They're just showing a little bit of variation in spine coverage. Those are falling out in this purely South American clade. And so this is a very clear long distance dispersal event from a close relative from South America into Cuba. Then we have this species Melocactus nagi and Pereza soy, which were thought to be part of the Harlowi group. It turns out they're not even close relatives of the Harlowi group. They're more closely related to things like Curvaspinus. And then we have what has been synonymized as Curvispinus guittarti, fallen in the Curvispinus group. So that makes sense. Holguinensis also fallen within this Curvispinus group. But you can also see we've got some of these other species, Melocactus macrocanthus, Caroli lenai from Jamaica, Broadwayi from the Southern Lesser Antilles, fallen within this Curvispinus group. So there's a lot of morphological variability in this group. These species are, can be pretty hard to tell apart. And so it does beg the question, what do we do with Curvispinus? Do you recognize it all as one giant variable species, or do you break these up into hard to distinguish multiple species in these different areas? If I show you, so I've got a photo here at the bottom of Melocactus guittarti, and that, if you show me a typical Curvispinus, that looks just like Curvispinus to me. But then you see something like Melocactus holguinensis here. Yeah, it looks like Curvispinus, but not super typical, right? And so therein lies the question, do we continue to recognize these things as separated out from Curvispinus, or as all part of a very highly variable species? So there's a lot of work there for someone to do. Okay, let's look at the Cuban clade. So we have these species, Harlowi and Acunai. Acunai is the one that I've photoed here, gorgeous plant. What we found essentially was Harlowi forms a more western distribution, Acunai forms a more eastern distribution of this species complex. We don't really see a lot of intermediates between the two. They're sister species, and so we made the decision to recognize both of those as separate species. But there was one Acunai, which was sometimes called subspecies lagunaensis, which was not closely related to typical Acunai. And so we raised that to the level of species and called it Melocactus lagunaensis. And it's actually closely related to this other species that occurs on serpentine called Rhadoxi. And then this other material that had been called Harlowi actually falls much more closely related to this multi-stem Melocactus evi. And so we describe that as a new species, Santiagoensis, from Santiago, Cuba. Okay, what about Melocactus pedrenalensis versus Melocactus intortus? You can look at these. These are photos I took of intortus on the island of Antigua. And this is, of course, pedrenalensis from southwestern Dominican Republic. They kind of look, they look similar, and they're both giant. Melocactus pedrenalensis also gets really, really massive. But it turns out they're not even close relatives to one another. You can see intortus here, and then pedrenalensis is way up, forming another part of the Caribbean clade. And so they're not close relatives. And so we should not consider pedrenalensis part of intortus, as it was named subspecies dominensis. Yeah, we should not call it that. So I want to make sure I'm not overgoing my time here, because I know we started a little late. What we can conclude from this work is the genus Melocactus in general is super young. So it's about 1 million years old, originating in South America. It's arrived in the Caribbean at least 4 separate times, most likely via long distance dispersal by potentially migrating birds or other means. That's something that we also have to spend a little more time thinking about and working on. We have these in situ radiations on both Cuba and Hispaniola. And what I mean by that is we have a group such as the Cuban clade, they're all endemic to Cuba, and they've radiated or diversified on the island of Cuba and essentially nowhere else, right? And so that would be Melocactus harlowi, Acunai, evi, Rhadoxi, lagunaensis, etc. And then we have those few species that have radiated on Hispaniola as well. We have about 10 species rather than just 3 from Cuba. And so this work has really helped us understand also what we need to do in terms of conservation priorities, because if we lump things super broadly into 3 species, instead of understanding their evolutionary relationships and showing now here with this work that we have 10 species, conservation priorities and strategies would be much more different, right? And this also just gives us a way to show more biological reality rather than the way that they were lumped into just these 3 species previously. The Curvus bianus group is in a tremendous need of population level work to really tease apart what's going on in this group. And to, like I said, do we call this all one species? Do we separate them out broadly into multiple species? It's going to be a tremendous project that someone needs to work on. And it has a really massive distribution too. So it's southern Mexico through Central America and northern Caribbean all the way up into the Greater Antilles, right? So it's going to take some really interesting broad scale work to figure that out. There are many more South American species that need to be included in phylogenetic analysis like this, and then nuclear data sets also need to be utilized, and that's something we're working on. We need chromosome numbers across this group because although we've been able to tease them apart with the phylogeny, if we had more information on chromosome number, it also might give us an idea of why we have so many species in Cuba, for instance. Is that due in part to hybridization and their evolutionary history, or what is going on there? So chromosome numbers are something we're actually working on at the moment to try to understand whether or not we have diploids or tetraploids, whether or not they've undergone genome duplication. So what about some of the future directions? We need to incorporate all of these Caribbean taxa into nuclear data sets with the rest of the South American species. And so Monique Homero Brito and other authors have been generating some really nice nuclear data sets for Melocacti. And so the idea, one thing that we're intending to work on now is including all of those species within those existing nuclear data sets, so we can have an absolute total picture of the diversification of this group across its entire distribution. We've been doing quite a lot of field work as well. This is a photo I took at Cañón del Pato in Peru last year. And you can see these really amazing Melocactus peruvianus sort of there growing along with Hagioserius. And it's a really curious group in Peru as well because of their, Melocactus peruvianus has a really wide distribution. And there's another species that's not a close relative, Bayuvistensis, that's much more restricted to tropical dry forest. So, and we did have both those species in our previous phylogeny, but we need to include, essentially all species into the next round. And that's something that we're getting ready to tackle. So there are a couple of missing Caribbean taxa that have not been analyzed yet in any way, shape, or form. So there's this material that was described by Hoxie et al., Melocactus calosanthus, which in our checklist, we actually considered this to be part of Pederneilensis. Because even when they described the species, they had very normal material Pederneilensis that they found growing directly with calosanthus. And so, and a bit of the morphology you see in calosanthus overlaps substantially with Pederneilensis. But it still would be really nice to be able to include members from those populations into a phylogenetic analysis so we could have, even more data to throw at this taxonomic issue. Then there was another new species described from Puerto Rico by Gedanik et al., Melocactus pygmaeus. And it's unclear what its most closest relative is. It actually looks like it could potentially be closely related to South American taxa, but they suggested maybe Lemidi, and I think Areces has suggested maybe Preripticula. And so it'd be really nice to be able to include this species into a broad-scale phylogenetic analysis, so we could really, sort of complete our existing knowledge for the Caribbean. So there's some, I would say relatively low-hanging fruit that need to be tackled so we can provide a much more comprehensive picture of Melocactus across the Americas. With that, I would like to, give a few acknowledgments here. There have been a number of agencies that have financed this work, and then a lot of people that have helped with this work as well. I wanted to specifically give a shout out to Lucas Bacci, who was a postdoc in my lab that helped with a lot of what we did in the paper, and Teodoro Clase, who's a colleague of mine on Hispaniola, who has helped with field work, and of course Edgardo Diaz and Daniel Barrios, which without them, this project would not have happened. So, so with that, I think I did okay on time. Just checking here.
You're golden.
Okay, excellent, excellent. Okay.
Let me see if there's anything on the Q&A.
Okay.
No. I had a question based on genetic studies. Are the genetics of the chloroplasts, you're talking to non-scientists now, different than the genetics of the nuclear material?
That's a fantastic question. That's a really, really great question. So in general, the chloroplast genome does not evolve as fast. The mutation rate is not as fast as the nuclear genome. And so I would expect in the nuclear genome, we'll get even more resolution than we did with the chloroplast genome. But one thing that can happen, because in many groups of, especially flowering plants, the chloroplast genome is inherited through the maternal line. And so oftentimes, if you just have the chloroplast genome, and if there's any hybridization, you're going to get only the signal from the maternal line, right? But if you have the nuclear genome, which is inherited from both parents, you're going to get, something, maybe a conflicting signal, or you might get just a totally different signal altogether, right? And so it is possible that if we analyze this with nuclear data, we might get a slightly different story than what we got with the chloroplast data.
Now, are there any constituent parts that have the male line represented in their genetic structures?
So the nuclear data does have both the male and female, and something I didn't mention because it's complicated, and we don't have enough data in Cactaceae yet. But sometimes in Cactaceae, the chloroplast is actually inherited through the male line. And and for the groups we have data for so far, it looks like a punch is almost always maternal. Looks like Cylindra punch is maternal. But there have been some other members of the Cactoides group, so where Melocactus would fall, where they've shown that it can actually be, maternal or paternal. And so that adds another level of complexity. That's also another, area of research that we just don't have enough information for, and it would be great for someone to tackle that and try to figure out what it is for different genera, let's say.
And since the Melocactus group is so young, maybe that's a recent development.
It could be, yeah, sure, it could be. Yeah.
There are some questions in the QA now. So you want to take them, Erwin?
Why don't you do it, Kelly?
Well, Stefan Campbell asked if wouldn't splitting Curvus spinus complex make sense for localized conservation efforts, but because when you consider it all as one, it becomes less fractioned, and sometimes the more endangered population gets overlooked.
Yeah, it's another really, really good question. I think that from a conservation aspect, that could be a really nice way to do it. So for instance, on Cuba, there's essentially just maybe two populations of what was called Melocactus guittarti, which has been lumped broadly in the Curvus spinus. Roughly two populations there, and they're not, they're invasive. There's this thing called marau, and it's this invasive legume that's coming in there, and it's really messing up the populations. And so from that point of view, I would say it probably would be better from a conservation perspective to split it up into multiple species. I think where it would get difficult is when you're trying to tease those things apart. Let's say you have them growing side by side in your greenhouse or something. How do you tell them apart, right, without knowing exactly where they came from? That's
the only reason data and the other genetic research is needed, so you can differentiate them.
Yeah, right. Exactly.
So just because two things look alike doesn't make them the same thing. There's another question in there. Does 40 to 50 degree kill Melocactus? And my attitude is anything below 50 degrees, they don't tend to like. So, but I wondered if you had some growing experience with that.
They don't like the cold, but I've never had one die below 40 or 50. Like I've had them go down to almost freezing and still survive. They don't like it.
They hate it.
But and the other thing, as you might imagine, they love humidity, right?
Yeah.
So here in Florida, I'm in North Florida, and when the rainy season starts in July and August, they start growing like crazy. They really, really love that. But yeah, they do not like the cold. But I've never had one die going below 40 degrees.
Yeah, they don't like, just from a grower's experience, they don't like cold wet. So if they are cold, make sure you keep them on the dry side. I would say that they're better grown as a greenhouse plant. For me, at least, we're here in sunny Southern California, and I have a couple of friends that have tried them outside, and ultimately an El Nino will take them down.
Lucas, has anybody tried hybridizing Melocactus with Arahadoa or disco cactus?
Ooh, that's a really good question. I honestly, I do not know. I bet you could form hybrids at least with disco cactus,
Mm-hmm.
but I do not know. I don't know. Yeah, I'm not sure.
I've seen a couple people try, and it doesn't result in fruit formation.
There
A
you
quick
go.
addition to the cultivation question here. A lot of the advice that you'll see about melocactus online is based on European growing advice. So don't let them drop below 59 degrees, which is conveniently 15 degrees centigrade, and that scares a lot of people off. In the US, I would say in most regions they're very easy to grow, as a lot of people here are saying, and so they're often overlooked in terms of collections, and they're really worth it. So grow melocactus.
Just no cold wet.
One question asks, how come there be 4 species of Melocactus on the tiny island of Aruba?
That's a really good question. That's a really good question. That's most of those. I mean, yeah, essentially all of those are forming part of that Curva spinus complex, right? And I think there's probably some really interesting, maybe even in their deep history, but some really interesting hybridization that's gone on within that group that has caused some of that speciation. And perhaps that's why you're getting so many species in such a small area. That's one of those places that would be really great to study those populations and try to take a stab at figuring that out, right? One other thing that can potentially happen, I'm not saying that that's what has happened in Aruba, but you could have had multiple, let's say, dispersal events from different parts of distributions of different species that all ended up in the same place, and if the habitat's right, that melocactus is going to grow there. So it's also another possibility given the location of Aruba and taxa within the Caribbean, taxa within Northern South America. It's a pretty good place for things to meet up.
There's another question here on the Q&A from Carol. Is it unusual for 2 areas of distribution, i.e. Cuba and Hispaniola and Brazil? Or did you suggest that Cuba came from South America and further diversified through breeding?
That's correct. That's correct. So you could say the most recent common ancestor of those groups that you find in Cuba are South American in origin. Yeah. But that in situ radiation that happened on Cuba is basically a diversification event that happened after they got there. So that what we call the Cuban clade is a diversification event that happened after that, let's say long distance dispersal event from South America.
There was one question about Melocactus guanensis, and that's a Colombian species, right? Guanensis. So where do spineless species such as guanensis fit in the genus complexes?
Oof. Yeah, that's a good question. I mean, one thing we have seen is that spine morphology and spine production is super variable. Even within a complex, you can get that sort of. That's there are no spineless entities necessarily in the Curva spinus complex, but essentially most of the morphological diversity you're seeing there has a lot to do with spine production. And things like Actinocanthus was separated out because of its lack of numbers of spines as compared to Matanzanos. And that's a pattern that you can get sort of over and over and over again due to localized adaptation. It's actually something that I've seen among populations of Melocactus peruvianus in Peru. can see some really drastic differences in spine coverage, spine production, depending on the sort of microenvironment or the habitat where you find them growing. And if you compare across, say, all the populations of Peruvianus, there's an incredible amount of morphological diversity there. And a lot of it has to do with this local adaptation to these microenvironmental conditions. And so even within the species, you could potentially get, the production of something that's, let's say, maybe not spineless, but almost nearly spineless, and something that has really robust spines, right?
I know when we found them in Colombia, we found them on cliffs and they were well protected. So not having a spine, I could see how it developed, much like, sightlessness in the dark cave fish kind of thing. It's not needed.
So that's a really interesting point you make, Kelly, because I've seen very similar things in, say, Melocactus peruvianus in tropical dry forest that's under quite a bit of shade and broadleaf shrubs and trees, and then you compare that with something that's out on western slopes that have no cover whatsoever. That change is incredibly different. Numbers of ribs, numbers of spines go way down whenever you have them in these shaded conditions rather than in the white out open. So I think that there's a lot to be said.
Like, not having spines is not a disadvantage if you're well protected, but not having spines can cease your existence if you're in an area where animals get you.
Yeah, that and just the the harsh environmental conditions from the sun's rays and heat, that can take the plant body and overheat it so much if you don't have the spines protecting the epidermal layer.
Giving its own shade. Yeah. Yeah. So anyway, it's interesting, and I would say it's really I loved seeing all the different relations there, and I think that it's some parts that we're missing in a lot of talks. So I appreciate what you did there. Thank you.
Oh, great, thank you.
And I just had one clarification point
Yeah.
between polyploidy and hybridization. Is polyploidy usually or a result of hybridization?
It's usually a result of hybridization, but not always. So there are situations, there are a lot of choyas actually that within one species will form a polyploid. So think about Cylindropuntia fulgida that has diploid and triploid population, or the teddy bear choya, Cylindropuntia bigelovii, has diploid and triploid populations. And so that's what we would call autopolyploidy. So it's a genome doubling within one species. And there's been a lot of research to try to figure out why that happens. It's not truly clear why it happens, but we think it has something to do with some sort of an environmental perturbation, an environmental disturbance, and which causes the gametes to not reduce down into a haploid level. And so you might have a gamete that has the same chromosome number as mom or dad. So it would have a diploid level. And then if that crosses with another diploid gamete, you get an automatic polyploid, right? But so that can happen within a population of one species. And so you could get a triploid or a tetraploid within one species. But so that happens a lot, but it's much more common when you get a cross between one species and another, because the two genomes aren't really closely related. And so to make the chromosomes function the way they're supposed to, if you double that number, then you can have a pair of chromosomes from mom and another pair from mom, and then a pair from dad and another pair from dad, and they'll cross sort of by themselves within that one cell. It's crazy. But that's why most of the polyploidy that happens is derived from hybridization. But you can get it without hybridization. It can happen.
And having that broad genetics, is that why there's sort of a hybrid vigor to some polyploids?
That's absolutely right. That's absolutely right. That's one thing we see really commonly across these groups that have polyploidy. Oftentimes, let's say the parental species with fewer chromosomes are kind of restricted to really nice habitat. But then if you go into places where you might think, I wouldn't even normally expect them there, and they do just fine, the polyploids do, because they've got this hybrid vigor, right? That's enabling them sometime to to be able to fight against these adverse environmental conditions. That's why you get apunches that go all the way to Canada, because they're polyploid and they can handle it. They can take it. Yeah.
So I would assume we would expect polyploid species or polyploid plants to handle climate change better,
I think in some,
typically.
They
yeah, I
have
think,
been. They have been for millions of years.
and yeah, a lot of places where we find polyploids and where polyploids have developed are places where in the Pleistocene, glaciers were melting, and then sort of you had this sort of ebb and weave of glaciers and pushing populations back and forth and creating lots and lots and lots of disturbance and adverse environmental conditions. That's where we find a lot of polyploids, actually. Yeah. That's one thing we're studying at the moment too, with regards to the Andes, because there's been a lot of glaciation in the Andes, also associated with the Pleistocene cycles, and we think that probably we'll see some evidence of polyploidy happening with those Pleistocene cycles.
And are there any more questions? Because I promised Lucas that I would try and free him early from Q&A so he can go celebrate a birthday with family.
We should mention that this will be on our website. It will be recorded, right?
Yes.
Okay.
Happy birthday to whoever it is.
Oh, thank you. It's mine. It was earlier this week. So
Oh, very cool.
Congratulations.
Happy birthday.
thank you, thank you.
Thanks, Lucas. It was very well done. Thank you.
Thank you very much.
Thank you so much for the invitation. Again, it was an honor to be here with you today.
Thank you.
Yep, thank you.
Everyone be well, and I hope I see you at the convention.